Now moving on to our second investigation, this one is focused on understanding how the concentration of carbon dioxide affects the rate of photosynthesis. I will take you through each section of this investigation and explain the entire process and what we learn from it. So, starting with the main title, this investigation is all about studying the effect of carbon dioxide concentration on the rate of photosynthesis in submerged aquatic plants. So the focus is on how increasing or decreasing the amount of carbon dioxide can influence how quickly or efficiently the plant carries out photosynthesis. And, talking about the objective of this investigation, the aim here is to see how different amounts of carbon dioxide affect the rate at which the plant produces oxygen. This is because oxygen is released as a byproduct during photosynthesis, so if more oxygen is produced, it means photosynthesis is happening faster. We also use a special solution called hydrogen carbonate indicator to help us observe changes in the level of carbon dioxide. Now coming to the experimental setup. This diagram shows all the important equipments that we will use. There is a light source like a lamp to provide light energy. Then we have a thermometer to monitor the temperature, an inverted funnel, and an inverted boiling tube placed over the aquatic plant, which is submerged in the solution. All of this is held in a beaker. This setup helps us collect the oxygen and observe the color change in the indicator. Next is the materials list which we will use. It involves an aquatic plant such as Elodie or Carbomba. These plants are ideal because they clearly release bubbles of oxygen, which we can easily observe and measure. Then we need the hydrogen carbonate indicator solution. This is important because it changes color depending on how much carbon dioxide is present. We also need beakers or test tubes to hold everything, a light source to provide consistent light, a stopwatch to keep track of time, a measuring cylinder to measure the solutions, and a thermometer to make sure the temperature stays constant. Lastly, we use a ruler to keep the distance between the light and the plant the same throughout the experiment. Now let us look at how we prepare different levels of carbon dioxide for this experiment. We will prepare several solutions using sodium bicarbonate, which dissolves in water and releases carbon dioxide. By adjusting the concentration of this chemical, we control how much carbon dioxide is available in each trial. We typically prepare a control solution with no carbon dioxide, and then other solutions with increasing amounts such as 0.5%, 1%, 2%, and 5% sodium bicarbonate. Now moving on to the experimental setup in more detail. We keep the temperature steady using a water bath, usually between 20 to 25 degrees Celsius. This is important because temperature can also affect photosynthesis, so we must keep it constant. The aquatic plant is placed into each prepared solution. The light source is kept at the same distance to make sure light intensity is not changing. Hydrogen carbonate indicator is added to each solution, and its color will help us observe carbon dioxide levels. At the start, the solution may appear yellow if carbon dioxide is high. As the plant uses up carbon dioxide, the color may shift to green or even blue. Let us now go through the procedure step by step. First, we place the plant in a beaker filled with one of the prepared sodium bicarbonate solutions. Then we turn on the lamp and start the stopwatch. Over a set time period, for example 10 minutes, we measure how much oxygen is produced. We can do this by collecting the gas in a syringe or using the water displacement method. At the same time, we observe the color change in the indicator solution. We repeat this process for each different carbon dioxide level, and we also repeat each trial 3 times to make sure our results are accurate and reliable. Moving to the observations. When there is no carbon dioxide, meaning in the control solution, the plant barely produces any oxygen because it does not have the raw material needed for photosynthesis. When carbon dioxide concentration is moderate, such as 0.5 or 1%, we start to see more oxygen being released and the solution may turn light green. At higher levels like 2 or 5%, the plant produces much more oxygen and the indicator turns darker green or blue, which shows that carbon dioxide is being used up more quickly. Now let us look at the results. There is a clear pattern. As we increase the concentration of carbon dioxide, more oxygen is produced, meaning the rate of photosynthesis goes up. For example, in a solution with 5% sodium bicarbonate, we see the highest oxygen output and the indicator turns blue. In contrast, at 0%, there is no oxygen produced and the solution stays yellow. This confirms that carbon dioxide availability has a direct effect on photosynthesis. Now let us understand the effect of carbon dioxide. From these results, we learn that increasing the amount of carbon dioxide increases the rate of photosynthesis. This shows us that carbon dioxide is one of the limiting factors. When it is in short supply, the plant cannot photosynthesize efficiently. But as we give more carbon dioxide, the process speeds up. And finally, a quick summary of the whole investigation. We designed this experiment to explore how carbon dioxide levels affect photosynthesis. We varied the levels of sodium bicarbonate to change the amount of dissolved carbon dioxide available to the plant. We measured the amount of oxygen produced and watched how the color of the indicator changed. By keeping other factors constant, we could clearly see how carbon dioxide alone affected the rate. As we increased carbon dioxide, photosynthesis increased too. But beyond a certain level, the increase stopped, showing that other environmental factors were now limiting. So the experiment helps us understand the idea of limiting factors in photosynthesis and the key role of carbon dioxide in that process.